All right. Hello, everyone. Good evening and a warm welcome to today's 1 hour exclusive free webinar session for all the science enthusiasts. Without wasting any second, we'll jump into the presentation as I appreciate your participation and I also respect your valuable time. Molecular docking has become a foundation of modern computational biology and drug discovery, enabling researchers to explore how biomolecules interact at atomic level, often before any wet lab experiment is performed. Yet for many students and early career researchers, docking still remains a concept that feels abstract, tool heavy, and difficult to approach with confidence. On a personal note, I have been in the field of bioinformatics for 8 years. In the last 3 years, I've been teaching postgraduate students on the topic of bioinformatics. It was then last year that I understood that what actually students really need is not actually motivation, but to be exact, real-time practical information for grasping such complex topics. Rather than giving boring lectures, if you need a student to listen to you, you need to give some sort of proof that the subject that you teach is actually a part of modern world. Because as far as I know, to understand molecular docking, you need to understand science. This webinar, Unlocking Protein Interactions, a beginner's guide to docking, is designed to bridge that gap, providing a clear, intuitive information and introduction to the principles of molecular docking, focusing on why we perform docking, how we do it, what docking can and cannot tell us, and how theoretical concepts translate into real-time computational workflows. Rather than overwhelming the participants with equations or software complexity, this session emphasizes conceptual clarity, practical interpretation, and best practices commonly overlooked by beginners. By the end of this webinar, you will have a structured understanding of docking fundamentals. You will be able to critically read docking results and be better prepared to engage with hands-on workshops and independent computational studies in the future. This webinar is planned as a pre-workshop session, laying the foundation for deeper exploration into molecular docking as a powerful dry lab tool for studying protein-ligand and protein-protein interaction in biological and biomedical research. Now, taking all your attention to the presentation, today's session is hosted by VirtualChem Labs, and my name is Dr. Nitish Roy PA. I work as an application specialist for VirtualChem Labs, an exclusive e-learning platform. Before we go into any further into tools or workflows, I want to pause briefly on something more fundamental, the way we think as a student, a researcher, a scientist. As students and researchers, every method we use is driven by two questions, why and how. Asking this every time we reach an impasse increases our what is called scientific temper and critical thinking. Here, the why defines the purpose of our study. Why are we docking this ligand? Why this protein? Why this software? Why should this interaction matter biologically or clinically? Without a clear why, docking becomes purely computational exercise with no scientific direction. The how defines the strategy. How do we model the interaction? How do we choose the binding site? How are the parameters and the scoring method selected? How do we interpret the results responsibly? Molecular docking sits exactly at the intersection of these questions. It is not an answer by itself. It is a tool that helps us translate biological hypotheses into testable computational models. Good docking studies do not start with software. They start with a well-defined biological question, followed by a logical computational approach, and finally, a critical interpretation of results. As we move forward in the session, I want you to think of docking not as a button-clicking exercise, but as a hypothesis-driven scientific method, one where we understand why you are doing each step is as important as knowing how to do it. To give you a clear sense of how the session is structured, the slide outlines 5 key topics. We'll begin with basics. Here we briefly revisit the fundamental concepts of molecular docking. What is docking? What problems is designed to solve? And the terminology you need to follow the rest of session comfortably. Next, we'll move into tools. The session introduces commonly used databases, docking platforms with a focus on understanding what each tool is used for rather than learning software in isolation. The third is significance, where we discuss why docking is relevant in each context, how it supports hypothesis generation, reduces experimental burden, and contributes to design making, drug discovery, and molecular biology. In fourth, we'll walk through the workflow. This is where we connect concepts and tools into logical sequences, from target selection and preparation through docking to analysis and interpretation. Finally, we'll conclude with some case studies. The session demonstrates how everything discussed earlier becomes together in a practical example, showing how docking results are generated, analyzed, and interpreted in real-world scenario. By this end of session, you should not only understand how docking works, but also why each step matters and how to approach docking studies with confidence. The basics of molecular biology. Before we move into tools and workflows, it is important to align on a few core concepts that form the foundation of molecular docking. At its simplest level, molecular docking tries to answer one question: how does a molecule bind to a protein? And how favorable is that interaction? To do this, docking software breaks the problem into four key components. First is the pose. A pose refers to the predicted orientation and conformation of ligand when it binds to a protein. Docking does not give us one single answer. Instead, it generates multiple possible poses, each representing a different way the ligand could fit inside the protein. To further simplify, for all those who have found the initial explanation confusing, imagine this. What do we do? What do we all do when a camera points us? We pose, right? So that the image then generated will have the best looking side of us when analyzed afterwards. So in terms of docking, poses represent different ways the ligand could fit inside the protein. The next is the binding or active site. It's also called as a cavity. This is the region where the interaction occurs. In some cases, the active site is already known from the experimental data. In other cases, docking tools identify potential cavities or pockets of the protein surface where a ligand could bind. It's called cavity search. In the following slides, I will discuss some of the softwares which could do cavity searches. Simply said, every protein will have a cavity, whether it's an active site or not. What makes it bindable to a ligand is the combination of specific amino acids making the cavity in the first place. A few of the images that I have given in the bottom of the slide, you can look at the pockets, the groove, the cleft where the ligand is bound to the protein. The third is a scoring function. There is a mathematical model which is used to estimate how favorable a given pose actually is, usually expressed as binding energy. The scoring function evaluates interactions such as hydrogen bonds, hydrophobic contacts, electrostatics, and steric complementarity. Importantly, this bonds is a value is an approximation. It helps us rank poses rather than measuring true binding affinity. Finally, we have the search algorithm. The algorithm is responsible for exploring all the possible ligand positions, orientations, and conformations within the binding site. Because this search space is extremely large, different docking programs use different strategies, such as genetic algorithms or stochastic sampling, to efficiently identify the most promising poses. Discussion of such search algorithms comes for a full day session, as these are very complex topics. Together, these four elements, pose, binding site, scoring function, and search algorithm, define how docking works in practice. Understanding these concepts is essential before interpreting docking scores or comparing results across different software platforms. To further simplify, every docking program will have a scoring function and a search algorithm, which it helps to approximate the values to real-time analysis. This gives us a basic as well as better understanding on what actually happens during a protein-ligand interaction inside the body. These are some of the major bioinformatics or docking tools Now that we understand the basic concept behind the molecular docking, the next logical question is, what tools do we actually use to perform these steps in practice? This slide summarizes some of the most commonly used bioinformatic and document-related resources that you will encounter in real workflows. We usually begin with structural data, and for that, the most important resource is the Protein Data Bank, or PDB, accessible through RCSB website. This is where experimentally determined protein and protein-ligand structures are stored. Almost every docking study starts by retrieving a protein structure from this database. Once we have a protein structure, we often need to inspect, clean, or analyze it. Platforms like Proteins Plus provide a web-based tool for identifying binding pockets, analyzing residues, and preparing structures without it requiring heavy local installations. For those who are working in more integrated graphical environment, Discovery Studio Client is a widely used software suite. It allows visualization, protein preparation, ligand preparation, and interaction analysis through a user-friendly interface, although it is commercially software. On the other side, ligand side, the major resource is PubChem, where it is a comprehensive database of small molecules, of all small molecules. From PubChem, we can retrieve chemical structures, download the ligand files, and obtain basic physical chemical properties that is used before docking. For protein-protein interactions and estimating the binding affinity, we have tools like Prodigy Server. They are not docking tools in strict sense, but they help estimate the interaction strength once the complex is formed. The PDB tools provided by LabShare are a collection of online utilities that assist with format conversion, structure cleaning, and chain splitting when there are multiple chains for a single complex protein that are essential but often underestimated by beginners. For docking itself, one of the easy-to-use server is CB-Dock2. There is an automated docking server that combines cavity detection with autodock data, making it particularly suitable for beginners who are learning how binding site identification and docking work together. When working locally, autodock tools are used to prepare input files such as PDBQT, define grid boxes, and manage docking parameters. Understanding autodock tool is even more important as if you use web servers, because many web servers operate on the underlying same principle of autodock. Finally, after docking, we need to interpret results. The protein-ligand interaction profiler, or PLIP, helps identify the visualization key interactions such as hydrogen bonds, hydrophobic contacts, salt bridges, and pi-pi interactions, allowing us to move from docking score to biological interpretations. Taking together, these tools form a typical docking pipeline, from structure retrieval through preparation and docking to analyze and to interaction analysis. You don't need to master all of them at once, but understanding where each tool fits is essential for conducting a meaningful docking study. From this onwards, I urge each of you to take down notes and note of the questions. If the questions that you have has to be answered immediately, you can click the raise hand icon and interrupt the session. We'll discuss it. Or else, if the question you have need to be discussed at the end, please type in as a message so that I can review it after session and proceed for discussion. At this point, it is natural to ask an important question. Why does docking actually matter? What is its significance, and why has it become such a widely used approach in modern biological and pharmaceutical research? First, docking helps us predict binding modes, meaning it allows us to understand how a ligand is likely to orient itself within a protein binding site. This spatial information is critical because biological function depends not only on whether the binding occurs, but how the interaction happens at molecular level. Second, the docking significantly reduces experimental cost and time. It's obvious, right? Instead of testing hundreds or thousands of compounds experimentally, which takes years and years, docking enables us to computationally screen and prioritize a smaller, more promising subset for wet lab validation. This makes experimental design more focused and efficient. Docking also predicts protein ligand interaction. Beyond simple binding, Docking provides insight to which amino acid residues are involved, what types of interaction are formed, and how stable those interactions may be under idealized conditions. Another key advantage in docking helps accelerate drug discovery. By integrating docking early in pipeline, researchers can move more quickly from target identification to lead optimization, reducing both development time and cost. Importantly, for beginners, modern docking tools make these workflows accessible. Many platforms now automate complex steps such as binding site detection and scoring, allows students and early career researchers to focus on interpretation rather than software complexity. Finally, docking allows us to evaluate key parameters such as binding free energy, ligand orientation, and interaction stability. While these values are approximations, they provide a rational basis for comparing computer compounds and form testable hypotheses. To summarize this information on the slide, docking is not a replacement for experiments, but it is a powerful decision support tool that guides experiments in more informed and cost-effective section and direction. Now that we understand why molecular docking is important and that tools are commonly used, let us walk through a typical docking workflow. This slide summarizes the complete process from target selection to result interpretation. The first step is identification of protein target, which usually begins with the biological question, such as identifying a protein involved in a disease pathway or specific molecular interaction or protein of interest. Once this target is identified, its pre-randomized structure is obtained, most commonly the protein data from the protein data bank or from its homology model if an experimental structure is not available. The next step is identification of its binding pockets or cavities. Proteins, as you know, they are not flat surfaces. They contain loops and the pockets where other molecules like ligands and peptides can bind. These regions may be known from experimental data, or they can be predicted computationally using cavity detection algorithms. Why binding pocket identification is important because selecting the correct binding pocket is critical, as it directly influences docking outcomes, ergo, the reaction of the target protein with the drug. Once the binding site is identified, we move to preparation of the target protein and ligand. The step is often underestimated, but it is one of the most important parts of docking. Protein preparation includes removing unnecessary molecules such as crystallography water, adding hydrogens, assigning charges, and performing geometrical corrections. Ligand preparation involves generating the correct protonation state, geometry optimization, and file format conversions. After preparation, we perform the docking step itself. Here, the ligand is computation placed in the binding of the binding site of the protein. Multiple process proteins of the proteins are generated, and docking can involve small molecule protein peptide interactions depending on the biological questions being addressed. The final step is analysis and interpretation. Docking does not end with a score. At this stage, we analyze binding energies, ligand orientation, and interaction patterns such as hydrogen bonds, hydrophobic and interfacial contacts, non-interacting surfaces, and electrostatic interactions. These results are then interpreted into context of biological relevance and used to guide experimental validation on further computational tools. In summary, docking is not a single-click process. It is a structured workflow where each step from target selection to interpretation contributes to reliability and usefulness of the final results. Identification of the protein target. The first and most critical step in any docking workflow is this. Everything that follows, binding site detection, docking accuracy, and biological interpretation, depends on choosing the right target. Protein target selection always begins with biological or clinical question. We ask the protein is it directly involved with a disease pathway, molecular mechanism, or biological process that we want to study. Docking should never be performed in isolation from biology. It must be hypothesis driven, simply meaning we can perform docking between any molecule and any protein, but there has to be a meaning to it. So it is said that Theoretically, docking should never be performed in isolation from biology. It must be hypothesis driven. We have to know if the docking will give a favorable result. Once the target is identified, the next consideration is structure availability, where we analyze the structure PTB, or pro-X-ray crystallography, NMR, or cryo-electron microscopic structures. If a high-resolution structure is not available, homology models can be used, but with additional caution. The next is identification of binding pockets or cavities. Once the protein target has been identified, the next crucial step is in the docking workflow is identification of binding pockets or cavities. This step defines where a docking will take place. Proteins are three-dimensional structures with grooves, cleft, and pocket formed by the folding in 3D space. Ligands typically bind with these cavities rather than with flat surfaces, making pocket identification essential for meaningful docking. In some cases, the binding site is already known. As I have pointed out, some of the pockets, cleft, grooves, and binding site in these 3D structures of protein, as you can see. You can use some softwares and web servers to do the identification of binding pockets if you have no knowledge or any of the literatures have never mentioned about the binding pockets. In some cases, this may be based on the experimental evidences such as co-crystal structures, mutagenesis studies, or biochemical assays. When such information is available, docking can be performed directly on non-active or allosteric site. And there are cases when binding site is unknown. Here, computational tool is used to predict potential cavities by analyzing protein geometry, structural surface topology, and physicochemical properties. These algorithms identify regions that are large enough and accessible and are chemically suitable to accommodate a ligand. It is also important to distinguish from functional relevance and geometric availability. Not every cavity detected by software is biologically meaningful. Some pockets may be transient, solvent exposed, or irrelevant to protein function. Critical evaluation is therefore required when selecting a docking site. The preparation of target protein and the desired ligand. After identifying the protein target and binding pocket, the next and arguably most important step in the docking is the preparation of target protein and the ligand. Many of docking errors originate not from the docking algorithm, but from the inadequate preparation. Starting with the protein, experimental structures often contain elements that are not suitable for docking. These may be included crystallographic water molecules, buffer ions, co-crystallized ligands, and that are irrelevant for docking study. During preparation, such components are carefully evaluated and removed if necessary. Protein preparation also involves adding missing hydrogen atoms, assigning correct protonation states, and defining partial charges. These steps are essential because most docking engines calculate interactions based on atomic charges and hydrogen bonding patterns, which are not fully present in raw PDB files. Equally important is ligand preparation. Ligands must be in the correct chemical form, including appropriate ionization and tautomeric states that reflect physiological conditions. This step ensures that both protein and ligand are chemically and structurally realistic before docking begins. Poorly prepared inputs can lead to incorrect poses, unrealistic binding energies, and misleading interpretations, even if the docking protocol is otherwise correct. Here, the preparation is not a technical formality, but it can be considered as a scientific quality control step. Careful protein and ligand preparation significantly improves the reliability, reproducibility, and interpretability of docking results. Once the protein and ligand are properly prepared, we move on to the docking step itself. At this stage, the docking is to predict how two molecules interact in three-dimensional space. Depending on the nature of the interaction partners, docking can be broadly classified into three types. Some of you might have already have knowledge about this. The three types are small molecule to protein docking, peptide to protein docking, and protein to protein docking, which is very complex. Low molecular weight ligand, when docked to a protein's binding pocket, is where we call small molecule docking. This approach is widely used in drug discovery to screen compounds, rank binding poses, and identify key interactions within an active site. Most beginner-friendly docking tools are optimized for this category. The second type is the peptide protein docking, where peptides are larger and more flexible than small molecules, which makes docking more challenging. Peptide protein docking is often used to study signal motifs, inhibitory peptides, and or short protein fragments involved regulation. Because of increased flexibility, specialized algorithms and careful interpretation are completely required. From the here onwards, the topic of docking with peptide and protein becomes a bit complex. So I'm not going to into that causes and to will cause confusion, and that causes this to explain such deep complex topics. We need at least a 3-day workshop. The third category is the protein-protein docking, where protein is computationally more demanding due to large interaction surfaces and conformational changes. It is commonly applied in studying signal pathways, immune recognition, and multi-protein assemblies. Docking is not a single method, as you know, but a family of approaches tailored to different molecular interactions. Understanding these distinct help in choosing the right tools and setting realistic expectations for docking results. The most critical step in all those processes is the analysis and interpretation. At this stage, we evaluate whether the predicted binding modes are biologically meaningful rather than just computationally favorable. During this time, I suggest, if you remember a few of the websites that I have already shown in the slides, you can just Google it or just look at the slide and understand which are the websites, and you can just Google it and understand how this site actually works. One of which is Proteins Plus, the other is Protein-ligand interaction profiler, the other is Prodigy. So you can go into that website and you can understand how this actually works, what molecules you have to upload, and what molecules you have to add to give such complex results. The first parameter typically examined is the binding affinity or docking score. Lower energy scores generally indicate stronger predicted binding, but scores should be used for relative ranking, not as absolute measures of binding strength. Next, we analyze the binding pose and orientation. Equally important is examination of molecular interactions. These include hydrogen bonds, hydrophobic contacts, salt bridges, pi-pi interactions. Identifying interaction with catalytically or functionally important residues increase confidence in the docking outcome. We also assess pose consistency and clustering. If multiple docking runs produce similar binding modes, the prediction is considered more robust. Highly scattered pores may indicate poor convergence and or an unstable docking setup. For a protein peptide or protein-protein docking, interface area, residue complementarity, and interaction networks are prioritized over raw docking scores. Finally, docking results must be always be interpreted in biological context. Docking provides hypothesis, not proof. Promising complexes should ideally be validated through molecular dynamic simulations or experimental approaches. To summarize this docking workflow set of slides, analysis interpretation transforms docking output into biologically actionable insights, bridging computational predictions with real-world molecular behavior. To summarize all, the workflow of molecular docking follows these 5 steps. Altogether, the workflow of molecular docking follows a logical sequence. It begins with the selection of a biologically relevant protein target, followed by identification of appropriate binding site of cavity. Then the protein and ligand are prepared to ensure correct chemical and structural representation, and it is achieved and then selected. Next, the docking is performed to generate multiple binding poses and estimate the binding affinity. Finally, results are analyzed and interpreted to identify meaningful interaction and prioritize candidates for further computational or experimental validation. Now that we have gone through the basics of molecular docking and docking workflows, we can go through some major case studies where docking was the major tool used to identify some of the drugs. All those theories that you have might wonder, what's the reality? If docking is so good, and the theory suggested if it's so cost-effective and time-accelerating factor for drug discovery, then here are some examples, real-world examples where molecular docking favored drug discovery. To understand the real impact of molecular docking, it is used. It is useful to look at how it has been applied across different stages of drug development, from early discovery to clinically proven blockbuster drugs. Let's begin with clinically proven blockbuster drugs. In this case, blocking is often used as retrospectively as a validation tool. Researchers used to use docking to rationalize why drug bind efficiently to its target. Here, docking supports mechanistic understanding rather than driving discovery. Moving to the blockbuster drugs, docking plays a more integrated and iterative role. For many high-revenue drugs, docking was used repeatedly during the lead optimization to refine binding affinity, selectivity, and pharmacological profiles. In contrast, the discovery phase and lead optimization case rely most heavily on docking as a decision-making tool. At this stage, docking is used to virtually screen large compound libraries, prioritize hits, and eliminate poor candidates early. The goal here is not perfection, but efficiency, reducing cost, time, and experimental burden by focusing only on most promising molecules. When we compare these three categories, where clinically proved, blockbuster drugs, or discovery phase, considering them as three, in discovery phase studies, docking is predictive and exploratory. In blockbuster drug development, docking is iterative and optimization driven. In clinically proven drugs, docking is explanatory or confirmatory. Importantly, docking alone never delivers a drug. Its strengthens lies in the guiding decisions, narrowing choices, and providing molecular level insight that complements experimental work. Molecular docking adapts its role representing depending on the stage of drug development and the characteristic limits in clinically validated drugs. To better appreciate the role of molecular docking, let's compare some case studies where we discussed earlier and understand how docking contributes differently at each stage of drug development. These two, one belong with the discovery phase studies and the other belong with the clinically proven drugs. As you can simply Google it and understand what these two molecules have been identified and how it has been identified and what the preference of docking was. The second set was these two. One was a kinase inhibitor and one was an influenza neuraminidase. Third one was an HIV protease along with a kinase inhibitor. Fourth one was a thrombin inhibitor, which was formed from the hybrid screening. And next one was a BCL and ABL. These are the set of drugs which were completely made or either completely made by docking or completely identified by the efficiency of docking itself. From classical bioinformatics to AI-driven dry lab drug discovery. For understanding docking, we are not supposed to skip how artificial intelligence has completely supported the mechanism of docking studies and modeling in the field of biotechnology and computational biology. In classical bioinformatics, we focus largely on sequence data, structural database, and rule modeling. Molecular docking, QSR, and homology modeling relied on predefined scoring functions, physical-chemical rules, and human-driven parameter selection. These methods were powerful, but they were slow, linear, and limited by human intuition. As we saw in earlier cases such as kinase inhibitor or renin inhibitors, docking primarily helped explain or optimize known interactions. As data grew larger and chemical space became vast, this is where AI and machine learning entered in the picture. In AI lab-driven dry lab drug discovery, algorithms learn directly from data rather than relying on handcrafted rules. Machine learning models can predict binding affinity, classical active versus inactive compounds, identify novel scaffold, and even suggest chemical modification during lead optimization. Deep learning models now integrate structure, sequence, interaction patterns, and experimental outcomes into undefined predictive frameworks. Most of you might know in 2024, the Nobel Prize for Chemistry was given to this software, this web server, AlphaFold, where the AlphaFold 1 version was released in 2018, and followed by 2 in 2020 and 3 in 2024. If you have been already using homology models, you can simply go for Swiss modeling and you can see that the database is completely hyperlinked to this AlphaFold server. Simply means when homology model is used to make a structure, model a structure, the structure's template is actually searched from AlphaFold database. So AI has completely taken over from this the world of computation biology also. Importantly, AI does not replace classical bioinformatics, but it builds on it. Docking still provides structural grounding, interpretability, and mechanistic insight. AI adds scale, speed, and predictive power. Together, they form a hybrid dry lab ecosystem where hypothesis generation, screening, and optimization happen largely in silico. The transition from classical bioinformatics to AI-driven drug discovery represents a shift from rule-based analysis to data-driven intelligence, turning dry labs into decision engines that guide smarter, faster, and most cost-effective drug developments. Our challenges as students and seekers. As learners and researchers, we face multiple challenges. The first is knowledge overload. Omics technologies generate massive data set, and distinguishing meaningful patterns is not trivial. The next is the interpretability gap. AI and computation tools inherited limitations. Most computation models use simplified physics. They approximate molecular interaction but cannot capture the full complexity of solvent events, effects, entropy, or allosteric changes. AI has become a core tool in genomics and proteomics. In genomics, it helps in variant calling, genomic annotation, GWAS prioritization. In proteomics, AI predicts protein structures, post-translational modification, interaction networks, and dynamics. However, it's important to remember AI cannot replace wet lab validation. A predicted phosphorylation site, for example, still needs experimental confirmation to know if biologically meaningful. The role of researchers will evolve. We will design experiments much faster and more efficiently guided by AI. Tools like AlphaFold can revolutionize your research. You can use it to predict 3D structures of protein lacking experimental data, analyze the effects and mutations, model protein-protein interactions using AlphaFold Multimer, provide starting points for docking studies or drug design. If you ask, can AI beat wet lab science? Currently, AI cannot outperform wet lab experiments. Complex cellular environments, post-translational modification, and emergent behaviors are still beyond computational reach. AI can infer functional basis on sequence, structure, or network context, but biology is context dependent. Function often depend on cell type, cofactors, or environmental conditions. To summarize, the whole presentation. First, we understood there are key basic concepts, four basic concepts to be exact: the post, binding site, scoring function, and search algorithm. Secondly, we understood that to validate online tools for every dry lab experiment, there are more tools actually than you can imagine. I have mentioned very few. If you can just simply Google, you'll understand that for each analysis, each type of analysis, which is dependent on either chemistry or biology of the molecule, you will have a lot and lot of web servers to analyze the analyze tools and to interpret results. Third one was the significance of molecular docking, where we understood that it can predict binding modes, it can reduce time, it can accelerate drug discovery process, it increases accessibility for beginners, and then the evaluation is what altogether creates the results. Then we went through the docking workflow, where target and protein pocket identification, target and ligand preparation, docking, analysis, and interpretation. We learned how to do it, how do we understand it, how do we predict it, how do we point it, and then finally, how do we interpret it. We went through some of the case studies. These are the list of set 1 and set 2 discovery phase clinical stage drugs, which we can simply Google it and understand how these drugs have been identified using molecular discovery, molecular simulation, and molecular interaction studies. Finally, when went through the, we discussed the involvement of AI. There are servers like AlphaFold 3, Molecular Operating Environment, and Achilles BDS, where you can actually use these servers, complete servers, without the use of a standalone downloadable software or a completely $100,000 laptop. You can just use the server if you have a speed internet connection and to understand everything for docking. For example, molecular operating environment is a multi-program server where you can do protein peptide docking, protein modeling. You can do immunological reactions, antibody-antigen reaction, structure-based design, and you can do a 3D modeling. You can do molecular dynamic studies, everything from a single server. So there are tools that you more than you can imagine, and you just have to find it according to what your need is. To finalize, this is our website. I urge you to go to our website for updations on our upcoming classes and workshops. There are two events which are upcoming. One is a 3-day online workshop on molecular docking, and the other is a online course in bioinformatics, which will be released in the mid or last of February, which involves a sophisticated syllabus designed especially for students and researchers who see complex questions, solve complex questions in both chemical and biological science. So I hope this 45-minute webinar have given you enough information to either start a new docking study or just to basically get the idea where to start. And I urge each and everyone to join the 3-day online workshop because more details will be discussed in that workshop, specifically blind docking via server and autodock. And we will use other chemical softwares to identify how a ligand is prepared, how the protein is prepared, how the binding site can be identified. Thank you. My name is Dr. Nitish Roy, and this program is completely supported by VirtualChem Labs. Thank you very much.
Thank you, Dr. Roy. If participants have
Thank you,
any
Bobby.
questions, they can ask.
We'll have a 5 to 10 minutes of discussion time. If there is any questions that you have, you can ask or just type in the message. I can see there is a message in the message box from Nobita. Uh, the question was, while selecting a protein or protein ligand interaction, the structure from database like PDB, what additional criteria should be considered apart from the biologic relevance? Simply speaking, if you are planning to get a structure from PDB, primarily you have to look for the resolution of the protein X-ray crystallography structure, which is submitted to the PDB. It should be at least less than 2 or between 1.8 and 2.2. The resolution increases to increase in number, like there are 3.5 angstrom, there are 3.2 angstrom. If you are particular about the quality of the protein, you have to go for a good resolution which is below 2.2 angstrom. And this angstrom is actually a point where this is done through identification of the length of the bond of each amino acid in the protein. So simply means the 1.8 angstrom resolution is how much you can actually zoom in to the protein. 1.8 angstrom simply means the length of a single amino acid. So that's what you can zoom in while you are doing the docking or analytical studies with that protein. Hope Namita understood the answer.